So the topic is urea cycle. Urea cycle is also known as Krebs-Henseleit cycle. Krebs-Henseleit cycle. The urea cycle, the urea is the product of protein catabolism. Protein catabolism. The site of urea cycle is liver. In the net reaction of urea cycle, for the production of 1 mole of urea, we require 4 ATPs. Then we require 1 ammonium ion, plus we require a nitrogen from aspartate. So alpha nitrogen from aspartate. So now let's proceed to the cycle. So to start with, we have nitrogen and carbon dioxide. So nitrogen and carbon dioxide. This carbon dioxide is derived from bicarbonate. And the ammonia is from the intestine, from the portal vein. Now this ammonia and carbon dioxide, the enzyme acting on them is carbamoyl phosphate synthetase. Carbamoyl phosphate synthetase 1, and this step is the rate limiting step. Of the cycle. We also require here 2 ATPs, which are converted to 2 ADPs, and we also require N-acetyl glutamate, which act as a catalyst here, which is also known as allosteric catalyst. So ammonia and carbon dioxide carbamoyl phosphate synthetase acts on it, and we have carbamoyl phosphate. Now let's take reaction further from this. I also wanted to tell you that the normal level of ammonia in blood is about 10 to 20 micrograms per dl. So now taking further the reaction, the carbamoyl phosphate plus the ornithine they act to form citrulline, and the enzyme acting here is ornithine carbamoyl transferase. As ornithine is involved and carbamoyl is involved, ornithine carbamoyl transferase, and we get citrulline here. This enzyme is also known as ornithine transcarbamylase. So it is also known as ornithine transcarbamylase. So ornithine carbamoyl transferase or ornithine transcarbamylase. Also, I forgot to tell that ornithine is formed in cytoplasm and then it is transferred to the mitochondria. So now we will continue from citrulline. The further reaction proceeds as arginosuccinate. And the enzyme acting here is arginosuccinic acid synthase. And yes, here ATP is consumed. It is converted into AMP, not in ADP. AMP plus PPI. And now this arginosuccinate proceeds as arginine and fumarate, and the enzyme here is arginosuccinase, as arginine is produced. Arginosuccinase or arginine, arginosuccinate, arginosuccinic lyase or lyase. So arginosuccinase or arginosuccinate lyase. We get products as arginine and fumarate. the fumarate uh, con- uh, the fumarate links the urea cycle to the TCA cycle. So this is important point. It links the urea cycle to TCA cycle or citric acid cycle, and the arginine continues to form ornithine and urea. And enzyme acting here is arginase, as arginase lies to ornithine and urea. And the one thing to mention is some of these reactions occur in the mitochondria of the cell, and some of this reaction occur in the cytoplasm of the cell. So this is the division. These reactions, that is the ammonia and carbon dioxide, then to the carbamoyl phosphate, then to the citrulline, this occur in the mitochondria. So I will write mitochondria, and here cytoplasm. Ornithine is synthesized in cytoplasm and then transferred here. So this is urea cycle. A quick recap: the ammonia and carbon dioxide, they are reacted by the carbamoyl phosphatase synthase, phosphate synthase, and then carbamoyl phosphate is formed. And carbamoyl phosphate, this is the rate limiting reaction here. Then carbamoyl phosphate and ornithine from the cytoplasm, they give citrulline, and the enzyme acting here is ornithine carbamoyl transferase or ornithine transcarbamylase. Everything right now is in mitochondria. Then the reaction proceeds to arginosuccinate, and arginosuccinic acid synthase is the enzyme. 1 ATP is consumed. Then we proceed to arginine and fumarate, and the enzyme here is arginosuccinase or lyase. This fumarate links the urea cycle to the TCA cycle, and from arginine we have ornithine and urea. Urea, the structure of urea is like this. Urea is highly soluble and non-toxic. Now we will discuss few disorders related to urea cycle. The first one is hyperammonemia. Hyperammonemia type 1, and the disorder is due to defect in the enzyme CPS, as we have seen, carbamoyl phosphate synthase 1. And the second disorder is HHH syndrome. And the enzyme involved is ornithine transporter. We have seen that ornithine is transported from the cytoplasm to the mitochondria. So ornithine transporter. And the third disorder is hyperammonemia type 2. Hyperammonemia type 2, and the enzyme involved is ornithine transcobalaminase or ornithine carbamyl transferase. So I will write OTC, ornithine transcarbamylase, OTC, ornithine transcarbamylase. Then the fourth one we have citrullinuria, and the enzyme defective is arginosuccinate synthase. Arginosuccinic acid synthase. Then we have arginosuccinic urea. And the enzyme defective here is lyase, as we have seen, arginosuccinase or lyase, which is also known as arginosuccinase. Then we have hyperarginemia, and the enzyme defective is arginase. One thing I forgot, that in arginosuccinic urea, the symptoms, one of the symptoms is tufted hair. So these are the disorders of urea cycle. Thank you for watching the video. Please like, subscribe and share the video.